there are several papers, and now comes to my mind two or three papers that Sophie Laillé and Olivier Manzoni in here in Bordeaux and in Marseille, in France, always. They clearly showed that omega-6 diets are leading to an increase of endocannabinoids and that increase the behaviors induced by endocannabinoids, that increase the electrophysiological effects of endocannabinoids. Yes,
I see.
It's much,
So,
but pretty much clear. Yes.
okay, so you just increase the omega-6 intake availability in an
Not
animal's
the only
diet.
way to increase endocannabinoids, of
Yep.
course. It's
Yep.
not the only way, but this is
Yeah.
a clear
Yeah.
one.
There's a direct link here. If you increase omega-6 intake in animals, they get more endocannabinoids. You see more activity, whether it's in the brain or elsewhere, you see things move in this direction.
I tell you more. We have now evidence. I don't tell you because it's not yet published the details, but there are specific populations that are specialized of cells of astrocytes in the brain that are specialized in the metabolism of certain lipids that are very excellent producer of endocannabinoids.
Hmm. So
So that
they
really
detect
that there is a
specific
kind of
lipids.
specific subpopulation of cells that are doing that job.
Interesting.
Yeah, this is
Hmm. So it's not so in your view, so as someone with this research perspective, it's not only is food, not only are calories generally abundant today, but the omega-6 polyunsaturated fatty acids are disproportionately abundant in the modern world. Do you
yeah,
think
yeah.
you think this is directly related to why we have an obesity epidemic and things like that?
As I said before, I don't have a proof. I don't know I should maybe ask Daniela, but I don't think that there is any human study that is really, yeah, there are human studies that are many human studies that are high fat diets, obesity is increasing endocannabinoids, all this, but there is a direct link. This I don't know, but it makes so much sense.
Right.
What we know is that if you eat too fat, your endocannabinoid system will be potentiated and probably you will eat more fat.
Mm-hmm.
So it is a self-perpetuating.
Yeah. And again, it makes so much
And
sense.
what are the molecular identities of this? There is some evidence, but not. I actually don't know the details, but
Yeah. But it makes
ideas.
so much sense when you step back and you take this evolutionary perspective and you think about time and the fact that for most of human history, most people were not in a stable food environment. Things were
Mhm.
seasonal, things were unpredictable, there were times of feast and famine. So it made perfect sense. There would be a survival advantage to any organism that had baked into its biology the tendency to keep eating as long as food was around, because it was inevitable that at some point it wouldn't be anymore. And if you didn't stock up and store with the future in mind, so to speak, then you would starve at a time of less abundance.
Yeah. And there was one thing that I wanted to add to what we were saying before, that these exostatic systems we were mentioning a little bit that expose you to dangers.
Mm-hmm.
For example, the one of eating too much.
Mm-hmm.
It's a danger. So what we found, and we and other people found, is that what we were saying before, that uh, um, very often, and this is probably the at the basis of this biphasic effects of cannabinoids, to do one thing or the contrary, it is a kind of safety system, safety mechanism for which, when the system is too much activated, there is a way to produce and to activate, for example, I can make you two examples of this. We found out that CB1 on excitatory neurons in the brain is promoting food intake.
Mm-hmm.
CB1 in GABAergic neurons, in inhibitory neurons, instead is inhibiting food intake.
Hmm.
And if you do a dose response, of an agonist of a THC, you will find that the increasing food intake, the munches of THC, are due to CB1 on glutamatergic cells. But when you increase the dose,
Yep.
even animals will stop eating.
Right.
And this is due to CB1 on the GABAergic cells.
Yeah. So
and this is also true physiologically. When there is too much endocannabinoids that are going, and what is interesting is that CB1 on glutamatergic cells in excitatory neurons is much more sensitive to endocannabinoids than CB1 on GABAergic cells.
So the initial release on the lower the dose or medium dose, you get the excitatory effect. If you keep going, you get the inhibitory effect. The whole system is well set up to sort of have this biphasic up or down, back or forth.
And another very, very, very interesting thing that again was something that I contributed with Pierre Piazza, but this is his discovery, is that there is a neurosteroid, a steroid that is produced in the brain that is called pregnenolone. Pregnenolone, that is a natural signal-specific inhibitor of CB1 receptors.
Yeah, this is important because most people don't know this about pregnenolone. So for those that don't know anything about pregnenolone, this is basically, it comes after cholesterol and before the other steroid hormones. We think of it as a steroid hormone precursor.